Dear students, today we are going to learn about binary fission. That is a method of reproduction of bacteria. Even though you know about bacteria, I just give a short, brief introduction. Bacteria are microscopic single-celled organisms found in our planet. They are the oldest life form and having a different diversity in shape, size, and function. The bacteria and also bacteria reproduce rapidly, allowing them to adapt quickly to changing conditions. So their reproduction is very fast. And also they play a important role in ecosystem and our human health and industry. We all already learned about all this. So we can take a look to what is binary fission. Binary fission is the primary method of asexual reproduction in bacteria. And in this process, the single bacteria cell, a single bacteria cell divides into two genetically identical daughter cells. That means a double clone of parental cells. A parental cell is converted into a double clone. One is converted into two. And we can see what are the key characteristics of binary fission. There is no meiosis and mitosis involved in this process. And in DNA of the bacteria or single cell bacteria, DNA of single cell bacteria replicates or doubled and cells place. Then produces clones of the parent cell or genetically identical daughter cells. This can be occur very rapidly, that means every 20 minutes in an ideal suitable condition. Next, we can see what the steps of binary fission, which is very important. So you should care about this. First step is DNA replication. DNA replication is the DNA of the bacterial cells duplicates or replicates. That means replicates. See, this is the single cell bacteria. This is called DNA. It gets replicates. It gets duplicated. See this. It gets two duplicates. There is two DNA. Okay. Then after that, a cell could become elongate, become long. That means size of the cell after replication, size of the bacterial cell increases. That is mean or cell elongates or gets longer and the size increases. Third step is chromosome segregation. The two copies of DNA move to the opposite poles of the cell. See in this image, the two formed DNA, identical DNA is moving into the opposite sides of the cell, elongated cells. That is called segregation, DNA segregation. Fourth step is cytokinesis. The cell membrane pinged inward and forming the septum that divides the cell into two identical daughter cells. See this picture. This is the a cell membrane pinches inward here and forming a septum inside and dividing a wall, forming a wall in between two DNA. And then it splitted and forms two daughter cells. These are the steps of binary fission. First one is DNA replication. Second one is cell growth and chromosome segregation and cytokinesis. You can be clear by this picture more clearly. This is the DNA, a single-celled bacteria, a prokaryotic parent cell initiates replication. Then when it replicates, there is a two DNA formed. A copy of parent cell DNA is formed. This is a genetically identical copy. This is replication of DNA. Then the cell wall, there is a septum formed in between the center of the cell, elongated cell. See the size of cell is increased here. Cell elongation is happening. Then cell wall inverts to pinches into inside of the cell, and there is a septum formed. That means cross wall is formed here. Then that is called and the DNA is moved into the opposite side of the cells. That is called by segregation, DNA segregation. Then the cells splitted into two cells and formed two identical and separate bacterial cell. That means two daughter cells identical to parental cell. So these two cells are identical or clone of the parental cell. You should care about this. I should remember that point. The two formed parental cells, the daughter cell, two formed daughter cells are identical to parental cells. Parent cell, that means these three cells are genetically identical, and these two are the clone of the parent cell. And for the conclusion, we can see what are the significance of binary fission. Binary fission enhances the rapid population growth of the bacteria. In this case, bacteria can double their number in as little as 20 minutes under ideal condition. I already said that in a favorable condition, within between 20 minutes, bacteria get double their numbers. And also genetic consistency, that daughter cells are clones of the parent, ensuring tight preservation. There is no change in the genetical conditions. And survival and adaptation of bacteria enhances. That means rapid reproduction helps bacteria colonize new environments quickly. That is also the new environment is similar to parent environment, the parent colony. In case of medical significance, we can understand binary fission aids in developing antibiotics and controlling infection. We can create in a controlled condition a similar population, genetically identical bacterial colony, and we can test antibiotics or developing medicines. In industrial application, it can be used in biotechnology, fermentation, and waste treatment by forming identical colonies, useful colonies. And this is all about binary fission.